Hello everyone, welcome to Chapter 13. This is Part 1. This chapter is all about the molecular basis of inheritance, and really we're talking about DNA structure and function. But we can start with a maybe simpler question, or taking a step back in time, is this idea: is the hereditary material DNA or protein? Now, we all kind of know it's DNA, but before this research was done that I'll just be describing in a moment, people didn't really know. In fact, we knew chromosomes were made out of chromatin, which, as you know, is DNA and protein. A lot of people felt that protein must be that hereditary material. Why? If you think about the number of amino acids that are found in a protein, you've got 20 different amino acids, right? Versus 4 different nucleotide bases in DNA. The prevailing wisdom was that, well, you can get a lot more information, quote unquote, in something that has, say, 20 letters versus 4 letters. So it made intuitive sense. Now, before we get into this question, it helps to know how DNA is structured. We've talked about chromosomes. We've talked about chromatin, which is DNA plus protein. But what are these proteins? Let's look more closely now. So DNA here, as we see in this nice figure, is wrapped around these histone proteins. So these proteins are called histones that are important for packaging DNA. And so you can go from DNA strand to what's called a nucleosome. This is this structure here with the histone and DNA wrapped around it twice. And this forms what's called this 10 nanometer fiber, or beads on a string. And you can actually see this by electron microscopy here. All right, so the histone DNA complex here, the unit of the nucleosome, it contains 8 histone proteins. So 1, 2, 3, 4, and then 4 behind it here, and the DNA is wrapped twice around it. And histone proteins tend to have a negative charge to them. DNA is a negatively charged molecule. Think of those phosphates giving it that negative charge. And so DNA is attracted to it, and then it wraps around it. So that's our early or lower levels of structure. We can go further. So we had those 10 nanometer fibers, those kind of coil up onto one another to form these 30 nanometer fibers, so a little bit thicker. And then these form these looped domains, which kind of stack in almost like a flower petal-like fashion. Sometimes you'll see this called rosettes. And then in a metaphase chromosome, when things are really getting packed tightly prior to cell division, these stack up even more on one another. So let's go back to this idea. We've seen this before, so this is a reminder. This idea that when a cell is just resting or actively just doing its thing, the DNA is packaged, wrapped around these histones. Some places or some parts of the genome are more tightly wrapped than others. And so the really dense or relatively dense parts are called heterochromatin. And you can see that in this EM here, the dark areas. Whereas the more loosely wrapped DNA, this is euchromatin. And if you think about gene expression, it's that euchromatin where genes are being expressed, right? Because the DNA is not all bound up and wound tightly around these histone proteins. And then, of course, again, during cell division, it wraps even more tightly. So this packaging of chromatin is dynamic. A gene can be effectively turned off by becoming more like heterochromatin, so much more tightly wrapped. And then vice versa, it can be loosened. So we see that here. So euchromatin, we see this unwrapping of the nucleosomes kind of spreading out, versus heterochromatin, they're very tightly packed. And any machinery that would need to get in there to express that gene cannot in the case of the heterochromatin. And we'll see how this works when we get into regulation of gene expression, the modification of histones, how they kind of wrap things up and unwrap. So even during interphase, or if it's the right kind of cell, that G0 phase, some parts of our chromosomes are still quite densely packed. Notable places would be the ends of our chromosomes and the what will become the centromere, that central part that we see during cell division. So that doesn't really need to unpack. There's no genes there that are needed to be expressed. So that's how DNA is packaged. Now let's get back to that main question. Is Well, what is that molecule of heredity? So testing to see whether this is DNA. And there are three major experiments that we'll go through. Let's get started on these with Fred Griffith's transformation experiments. So here's Fred Griffith with his dog, and he was studying Streptococcus pneumonia. He was a microbiologist, and he had different strains of this bacterium. And here are a couple of examples that we're going to focus on. This R strain, so it was named for because when you grow it on a culture plate, the colonies are not smooth, they're rough. And this S strain, so there were two different strains that he was looking at. This S strain was highly virulent, so you say put it into an animal and it will cause mortality, whereas the R strain is benign. So that's really important here. So let's see how that works. We'll take our S strain, inject it into a mouse. The S strain has this polysaccharide coat, and it will kill the mice. Now, if you take the R strain, this is this live non-virulent strain, inject it into a mouse, it's not virulent, it won't kill the mouse. Here's happy little mouse. What if we take the virulent strain and boil it? That effectively kills it. Inject that into the mouse, nothing happens. The mouse lives because you've killed the bacterium, right? It seems obvious. Here's where it gets interesting though. Take your heat-killed bacterium, which you know will not kill mice, mix it with a live R strain, the non-virulent strain. Now inject that into mice, the mice will die of the strep infection. So what's happening here? So here's what Griffith concluded. The R strain, that non-virulent strain, is picking up material from the dead S strain, that dead fragmented S strain. And Griffiths called this transformation. And we're really left with the question, of course, is, well, what is that information? So what can you say? What can we conclude at this point? All right. Well, we can't conclude much. We know something is being picked up from those dead fragmented S strains by the R strain, conferring virulence on it, but we don't really know what that is. So let's bump forward in time a little bit here and look at Oswald Avery's research. He picked up where Griffith left off. Unfortunately, Griffith actually died at a young age. And so Avery picked up on his research. And what his research group did was this. They did those same experiments like Griffith did, but they systematically eliminated one of the macromolecules in each of their experiments. So they heat kill an S strain and then treat it, say, with DNase. That's an enzyme that will destroy DNA. So you're selectively removing DNA from that particular prep. You do another experiment, and next time you remove all the RNA, for example. And so here's what that looks like. So we've got our virulent bacteria, you heat kill them, divide that up, you treat it with these different enzymes, or untreated RNAs, protease, DNase. You're systematically eliminating these different molecules. And so you mix those together, and what do you end up with? This is in this line here. You end up with virulent bacteria, virulent bacteria, virulent bacteria. The only time you eliminate that virulence, you're destroying that thing, that factor that the R strain is picking up, is if you eliminate the DNA. So this is very convincing evidence now that it's DNA. It's not protein, it's not RNA, it's DNA that's the molecule of heredity. Adding to this were Martha Chase and Alfred Hershey in 1952. They supported this idea that DNA is the molecule of heredity, and they also showed that DNA can effectively program cells, as we see with their research on these bacteriophages. So a bacteriophage like you see here, maybe you've seen cartoons like this before, these are viruses that infect bacteria. And a handy thing here is bacteriophages are only made out of protein and DNA. So we're eliminating other potential macromolecules, say like RNA, that could be involved in heredity. Now, a key thing, as we see in this electron micrograph, here's a bacterium, and all on the surface, here are the phages. So the phage is attached to the outside of the bacterium. We can go here. and they pass something, again, presumably DNA at this point, they pass something into the cell, and that is what programs the cell to then crank out a bunch of viruses. So bacteria will fill up with viruses and boom, the cell will explode, releasing tons of these new viruses. And so the question is, well, what is that information? We think it's DNA, but can we demonstrate that? So really cool experiment that they did here was they took two kinds of radioactive viruses. So they grow up viruses in cultures. They have their E. coli culture, that's the bacterium they're working on. And they have a bacteriophage that can infect their bacterial culture cells. And so they set up two different experiments, or two different cultures really. One that's grown in the presence of radioactive phosphorus, and so that's going to incorporate in the DNA. And then one with radioactive sulfur, where that's going to be incorporated into the protein, because think cysteine, that's an amino acid that has sulfur in it. And DNA doesn't have sulfur. All right, so they can then see, all right, we've got our nucleic acid radioactive viruses and our protein radioactive viruses. What gets into our bacterial cells? Because whatever is getting into the cell, that's that molecule of heredity, that's what's programming these E. coli to make more viruses. And so what they can do is, well, let the bacteriophages infect the cells, and then they separate those loose protein coats, those empty protein coats, and the bacterial cells, and they centrifuge them. And so the bacterial cells will go to the bottom of the tube. Just think, 10,000 times gravity, that's going to pull those bacteria down, and then those empty phages kind of float up in the medium here, this liquid. And so what did they find? When they have DNA, radioactive DNA, it pellets with the bacteria. That means that's what's getting into the E. coli. When they have radioactive protein, that's what we see over here, it doesn't pellet with the bacteria. That's not the thing that's getting into the bacterial cells. In fact, it just floats around up here. It's just the empty husks of those radio-labeled bacteriophages. And so this really hammered home the idea DNA is the molecule of heredity, and that's the thing in the case of bacteriophages that's getting into those E. coli. Okay, so here's a challenge for you. So a science fair project doing something similar to the Hershey-Chase experiment, but these students here substitute in radio-labeled nitrogen. Why wouldn't this work? All right. Yeah, so this wouldn't work because both proteins and nucleic acids have nitrogen. So that radio-labeled nitrogen will get incorporated into both, and you won't be able to distinguish them. So that wouldn't work. You need an element that's found in one but not the other. All right, so here's another question. What does DNA look like? I mean, could be a really broad kind of question. I mean, one way to answer this would be to do something like this. So check this out. Anybody, you could do this at home with the right ingredients here in a blender. Extract DNA from, say, a bag of frozen peas. This is pretty cheap. You need some soap. That's going to disrupt the membranes of the cells. Meat tenderizer breaks down the protein. You need some alcohol, isopropyl alcohol or ethanol, to precipitate the DNA, and you'll get this kind of gloppy substance. That's DNA. So there's one answer. DNA looks like that. It's not that informative from a structure standpoint. We could use electron microscopy. Here's what DNA looks like by EM. Again, we see it's this long, thin-looking thing, thread-like thing, but we really want to know more. So let's dig into that more deeply. There are a couple really important pieces to this puzzle of what does DNA look like. One is this work of Erwin Chargaff. He came up with these Chargaff's rules, and he studied all kinds of different organisms looking at the amount of adenine, thymine, cytosine, and guanine. Remember, those are DNA bases. And what he found consistently was, well, there are differences between different organisms, so that's interesting. It's not just universal, same percentage of any of these. But the thing that was consistent was he always found the same amount of adenine as thymine and the same amount of cytosine as guanine. So this is a really important clue. Another critical piece of information was generated by Rosalind Franklin and Maurice Wilkins using X-ray crystallography. So this is a technique which basically takes a compound of interest and you use X-ray diffraction to get basically a pattern from this crystallized substance. And you can interpret the structure or components of the structure based on that. And so Rosalind Franklin produced this figure, which has become very famous here. And this figure tells her that this has a helical structure. It also tells her some other information too, which we'll get to in a second. But back to Chargaff's rules. If you've got 20% adenine in the DNA of some species, what's the proportion of guanine? All right, yeah, it's got to be 30%, right? Because so if adenine is 20, that means thymine 20. That's 40% of the total. You're left with 60, which has to be split between G and C, which are equal. So 60 divided by 2 is 30. All right, so the other piece of information that Rosalind Franklin concluded, not just the helical shape, but that this DNA had 2 sugar phosphate backbones, and the nitrogenous bases were paired on the molecule's interior. Now, this was very important because as people were trying to figure out the structure of DNA, they had the sense of the sugar phosphate backbone, but were the bases oriented outward? Were there like 3 different strands of DNA? Were they all oriented outward? I mean, these were just things nobody knew. So her work was just critical for figuring that out. And so Watson and Crick, who are famous for ultimately elucidating the structure of DNA, Watson proposed that these DNA strands ran anti-parallel. Now remember, our DNA or RNA, nucleic acid in general, has an endedness to it. There's a 5' end that's based on the carbon here on the sugar group, and a 3' end. Here's the 3' sugar and a 3' hydroxyl on this 3' end. And so you have polarity, endedness. The strands could run parallel. They're both 5 to 3', say. Or they're anti-parallel. One goes this way and the other strand goes the other way. So this was a big step, this idea that Watson had that these run anti-parallel. Let's remind ourselves about the bases. Here are nitrogenous bases on their nucleotides. Here are the ones in DNA, adenine, guanine, cytosine, thymine. Those are the ones we're interested in here. And remember, they're different shapes. So the purines are bigger than the pyrimidines. And so if we're putting them together and using Rosalind Franklin's data, if you go purine-purine pairing, which was initially something that Watson and Crick thought, it's too wide. It doesn't fit her model. Same with pyrimidine-pyrimidine. That's too narrow. And then here's like the Goldilocks zone. Purine and pyrimidine, they fit just right and they match up with her data. So here's the base pairing that was proposed. And remember, this fits too that important key piece of information from Chargaff, that the amount of adenine is always the same as the amount of thymine. And it makes sense if they're base pairing, right? So for every adenine, you have a thymine. They're going to have the same proportion. And then the same for guanine and cytosine. All right, so ultimately, Watson and Crick put together this structure, literally put together a model of DNA structure where you have those bases oriented inward, the anti-parallel sugar phosphate backbones, and this is a double helical structure. So that's that helix, that was important, but it's a double helix. There's 2 strands. Now, just a few quick reminders. Nucleic acids are our building blocks in DNA. Here's that phosphate group. Sugar, in the case of DNA, it's a deoxyribose sugar. This nitrogenous base here, which ACG or T. And then we put these together, and when we do that, we form this phosphodiester bond, and we end up with a sugar phosphate backbone. Sugar, phosphate, sugar, and there'll be another phosphate here. And new nucleotides are only added on this end. So the strand grows from 5' to 3'. So what's keeping these strands together? Well, it's just hydrogen bonding, which goes back to when we talked about chemical bonding, how hydrogen bonds maybe aren't the strongest bonds in the world, but they might be very, very important. And that's very true here. So you have these really long stretches of DNA, a single strand, sugar phosphate backbone, bases extending outward, and this base pairing occurs between them. Notice you have 2 hydrogen bonds between T and A, and 3 hydrogen bonds between G and C. So actually, the GC bonds are a bit stronger than the TA bonds. And notice our strands are anti-parallel to one another. This one's 5' to 3' this way. This guy is 5' to 3' going up. Another force that's important for stabilizing this double helical structure are the van der Waals forces, or van der Waals interactions. Now remember, these were even weaker. These are further down the list in terms of those chemical bonds and interactions. But this is also called base stacking. So as this double helix twists, so we take our cartoony version over here and twist it into our double helix, notice the bases do get quite close to one another, and there are interactions between the bases. So again, base stacking. Imagine here's a layer of bases, here's another one, etc. So there are interactions between those as well. You'll see all kinds of models of what our double helix looks like. This kind of twisting strand, these twisting strands here is something you're probably familiar with. We can represent them this way, where we're sort of looking at the bases very block-like, and we still see a sugar phosphate backbone. Here we sort of ignore the shapes of the sugar phosphate backbone and just note what the bases are. Here's another way to show it. Maybe we just draw lines and note that this one's 5 to 3 down, this one's 5 to 3 up. And so there's many different ways to show that. Here, just a reminder, anti-parallel, so 5' to 3', 5' to 3' this way, but the bases match up. So G goes with C, T goes with A goes with T, etc. Hopefully you're getting the point. All right, so here was something that Watson and Crick wrote in their paper, and I always find this interesting. I almost think they're laying claim to some extent this idea that they're showing how clever they are. They know how DNA is likely replicated. So they said, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism. Now, they ultimately, as we'll see, didn't nail this down, but they were right. And here's what they're saying. You have this base pairing situation held together by hydrogen bonds. You can separate the 2 strands pretty easily. So here we separated them. Here, strand this one, and then this one. And now each of these can act as a template to make a complementary copy. Because an enzyme comes along and says, oh, here's an adenine. Well, we need to plunk a thymine there to match up with it. Here's a cytosine. We need to put a guanine. And so this idea had not escaped their notice. But let's see how this was figured out. So Mieselson and Stahl in the later 1950s studied 3 possible mechanisms. I like this. This is very scientific, although they probably had a good inkling what was happening here. They didn't rule anything out early on. And so there were 3 possibilities that they proposed. One was this conservative model that you have, that we're going to note the old strand or parental strand is in blue here. The conservative model means that you copy somehow the parental strand and you end up with a double helix of brand new material. So here's our old one. It just got copied and then say came back together. And we have another complementary copy over here. Semi-conservative, what's happening here, this is like what Watson and Crick proposed. Let's separate the 2 strands and then just fill in the matching bases on the other strand. So our new strand is complementary to our old strand in our new double helices. Another possibility was, well, maybe it's kind of a mix. Parts of this are copied and chunks are moved around, and here's some new stuff and here's some old stuff, and ultimately you end up with 2 exact copies, but it's some mixture of how this works. And again, if you think of this mechanistically like Watson and Crick did, this semi-conservative, like, just logically makes the most sense. It's easier to come up with a mechanism to how this might work. These other two, a little more difficult. All right, so how did Michelson and Stahl figure this out to demonstrate that this was true? You know, it's one thing to say, well, that semi-conservative kind of makes sense to me. It's another thing to actually show that this is true. So what did they do? They took E. coli. So a lot of this model research is using E. coli because it's easy, it grows fast, it's well known, and all sorts of people are using it. We have all sorts of genetic tools for studying things like E. coli. So we're going to grow E. coli in a medium that contains nitrogen 15. So that's heavy nitrogen. That's different than nitrogen 14, which is the sort of regular nitrogen, so our non-isotopic nitrogen. So we're going to grow our cells in this heavy nitrogen first. What that means is those cells with that DNA that has the heavy nitrogen, those cells are going to be a little bit heavier than, say, a cell that was grown in nitrogen 14, because the DNA is a bit heavier because they have this nitrogen with the additional neutron. All right, so we've got our heavy E. coli cells. Now let's move them into nitrogen 14, so a regular medium here. And then we'll see what happens to that heavy DNA after, at time 0, after 20 minutes, which would be 1 round of replication for E. coli, and then 40 minutes, which is 2 rounds of replication. And we can predict what might happen given our different models. So here in the conservative, starting in the middle here, but this was a first option earlier, conservative replication, heavy nitrogen DNA, we're going to copy it. And remember, the conservative means we conserve our original parent strand. So we'd really end up with 2 kinds of DNA. We're going to have cells with light DNA, nitrogen 14, and cells with the heavy nitrogen 15. There's no mix. We've conserved this one version and then just made copies of this lighter stuff. And then after 2 generations, this would continue down the road. We end up, we're just diluting our heavy DNA. In semi-conservative, again, we're just separating the 2 strands and then making a complementary copy. So you'd end up with a hybrid where it's half heavy, half light DNA. And then this next generation, you're just diluting that. So you're still got some strands that are heavy, but now you have some that are, some DNA molecules that are completely nitrogen 14. The dispersive would be kind of random. It's all over the place in terms of which parts are light and heavy. And so you're going to get a mix of resultant molecules. Now, how do they know whether they've got light or heavy DNA in their cells? They use centrifugation. So what centrifugation is you put your sample in a tube in a solution that will form a gradient where things that are heavier or more dense will travel further down in that tube versus things that are lighter, they're going to float at the top or not travel as far down in that gradient. And you apply that high speed, so 50,000 times gravity or whatever it happens to be for this particular experiment, and we separate out those strands or those molecules based on their weight. And so here's the result they got. So here's at time 0, separating things out. Things that are further south here, further down, are heavier or more dense. And so at time 0, all the nitrogen, all the nucleic acid is heavy nitrogen. That makes sense. That's what we would expect. After 1 round of replication, they found an intermediate level of DNA, which was not heavy, not light, but somewhere in between. So we go back to our conservative model here. What we would have expected there was half would be heavy DNA, half would be light, but we didn't see that at all. We've got some intermediate amount. So even after 1 round of replication, we can cross off that conservative model. Now, we still can't rule out the dispersive model until the next round. So based on our prediction here of the next round, or here are the results rather, half is intermediate or hybrid, heavy and light DNA, and the other half is light DNA. So given what we would expect from our semi-conservative model, that's what matches up. The dispersive model doesn't work because here we'd expect some sort of intermediate weight, so maybe somewhere in between these two lines here on our resulting centrifugation here, but we didn't see that, so we can rule out the dispersive model as well. All right, let's see how you do interpreting some of these results. So here's that Michelson stall setup, and so this is asking what would be expected at the beginning of the experiment before transferring their bacteria into the N14 media. All right, yeah, it would all be the heavy nitrogen DNA, so D would be the answer there. All right, let's try another one. So same setup, but different question. Which centrifugation would result in the figure above here would be expected after 2 rounds of replication if dispersive replication were occurring? Which of these would be consistent with that? Right, so that would be E, where you've got this kind of intermediate amount of heavy DNA. You don't have just light, you don't have just heavy, and then you're not quite in a halfway point, but somewhere in between light and half, hybrid. All right, so we can conclude that DNA is replicated by that semi-conservative model as Watson and Crick predicted, and each strand acting as a template for the other strand, which is somewhat just elegant and simple. And I encourage you to check out some of these resources here to see this in action, and that's what we're going to get into next. See you in the next section.